Welcome to Ask the Expert, everyone. This sort of format, the Ask the Expert, is a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording the event. We're going to post it on the Sugar Science YouTube channel shortly after presentation. And if you have any questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. Just a quick little bio sketch about Dr. Bender. He began his scientific career at WashU, a very beautiful place in St. Louis, where he received his PhD in developmental, regenerative, and stem cell biology. And then in 2015, he joined Dr. Chris Hughes' lab at UCI, beautiful sunny Southern California, to develop this microphysiological system, commonly referred to as sort of organ on chip. And it's aimed at cultivating human pancreatic cells in the lab. So you can just sort of imagine that if you can kind of have this system in place, you can do a lot of really great experiments to examine how islets behave in different circumstances. And ongoing work on this platform is aimed at establishing a model, right, to better understand how islets are damaged in type 1 diabetes. So and this is a question that's really never been solved, like what happens during the, on the way to presenting with full-fledged type 1 diabetes. So welcome so much for joining. I was appreciative that you're joining us, and just wanted to ask you, Dr. Bender, can you give us a quick thumbnail sketch of your career path and what really excites you most about what you're working on now, and talking about your paper, most recent paper and work?
Yeah, so thanks for the invitation. It's nice to chat about this work. It's been a lot of work that we've put in over the last 5 or 6 years developing this model, so we always like talking about it. My career really began in stem cell biology at WashU. So there I was working on brain stem cells and looking at how we can modify those cells with overexpression of certain genes and get either a healthy brain or a diseased brain. So it was really interesting work, very molecular. And at the end of that, I decided that I appreciated the molecular work, but I wanted to get at something a little bit higher level, something along the lines of cellular level and organ level. At an even higher level, I was interested in something that would allow me to apply my research more directly. So as something that has applications in biotech and the biomedical field. So interviewed at a few different labs here in Southern California, came across Chris's lab, and what I hope to convey today is that feeling that I had when I first saw this vascularized microorgan. I mean, just how cool it is that we can put these blood vessel forming cells in a platform, and through really minimal manipulation on our part, they form this beautiful biologically directed human capillary network. So it was immediately apparent to me that there were a lot of applications there. And so it was a great opportunity when Chris kind of said, well, I want you to take over this diabetes project. I had no background in diabetes whatsoever, so also an engineering component to our research. But I think I've enjoyed that. I've enjoyed the crossover between engineering and biology, and in a field that I've never really been involved with before. So that's kind of my research journey.
Yeah, I think that's interesting that you came to it with sort of fresh eyes. You're bringing all these tools to bear, but you weren't in the sort of T1D space. So I think that's pretty powerful.
Yeah, it's been interesting because there's I think we'll talk about today, a lot of stuff that's been done in the type 1 diabetes field hasn't changed that much in the last 20 years. So I don't know, I'm sure that every field has that to a degree, but it was more so than I'd seen in other fields. So it's interesting, and there are a lot of opportunities there to kind of make those next big innovations.
Yeah, there's a lot of very exciting work coming out of UC San Diego, UC Irvine right now, that we've been talking to different sort of players in that space. And there is really a lot of exciting, both engineering and genetic work coming out of both those institutions.
Yeah, absolutely. I agree.
So do you want to share a couple of slides about what you're up to these days?
Yeah, let me pull up my slides here. All right, can everybody see those?
They look beautiful.
All right. Great. So I'm just going to talk a little bit about the work we've done for the last 5 years. It's really been two-tiered. The first was just generating what we call an islet on a chip. And then more recent work, which I'll touch on only briefly because this is still very much in progress, is how we use that same platform to model type 1 diabetes. So I'll be wearing my UC Irvine hat today. That's where all this work has been done. But so that I am upfront with everybody, as the conflict of interest office likes me to say, I am also partly employed at Arcari Biosciences. This is the company that has spun out of Chris Hughes's lab, and but I won't be talking about that directly today. I'll only be talking about my work in the Hughes lab at UCI.
Sure.
So I mentioned that the diabetes field hasn't really changed all that much. And so for years, the way we've done islet research is we take pancreas, we digest it, we take the islets out, and then we grow them in suspension culture. So that's not physiologically representative for a lot of different reasons. One, when we isolate them, you actually rip off a lot of the supportive extracellular matrix. The pancreas is highly vascularized where islets are. That's important for supporting these organoids. Those are ripped off as well. Then after we've abused them like that, we let them grow in a suspension culture in a medium that I would argue is not really even physiological. There's a lot of different formulations out there, but in general, a lot of them have a much higher glucose level than you might expect to see at resting state in the body.
That's a really interesting point.
Yeah. We were kind of blown away by that. I talked about things that you kind of have a fresh perspective, but we kind of were shocked when we saw how much glucose there is in all these solutions, and how we're supposed to measure insulin secretion out of that is kind of head scratching. So we thought that there was a better way to do this, primarily so that we could facilitate better study of the islets in the lab. Plenty of mouse studies, but we can get into why that's not really the best model. So I mentioned the ECM and the blood vessels. So quite simply, we wanted to put the islets in an environment that recapitulated the ECM, regenerated that ECM, and then where we could also have blood vessels that could supply the nutrients and, most importantly, glucose to the islets, and then hopefully measure insulin that these islets would produce. So to do that, I want to introduce you to our technology that we've developed in the lab called the Vascularized microorgan. So these are microfluidic platforms. To give you an idea of the dimensions, the top to bottom of this schematic that I'm showing you here is about the thickness of a dime. And then the actual Z thickness of this is a fraction of a millimeter. So it's a very small tissue, but it's very powerful in what we can manipulate. Obviously, we do this so that it's easy to image and actually study the biological processes that are going on.
Can I just interrupt for a second?
Yeah, absolutely.
The other day we were on the State of the Science, and Peter Butler was on there from UCLA, and he was saying a few times, reiterating, you only need 500 milligrams of beta cells, right, to replace. That's sort of like, that was his marching call. But I think, I'm wondering how many milligrams of beta cells would fit into this chamber?
Yeah, so that's a great question. I don't know in milligrams, actually, that's not a scale that I usually work at, but we usually fit anywhere between 15 to 20 islets in these chambers. The one we actually use is a little larger than this. I'm going to show that on the next slide. But 15 to 20, we find, gives us the power to actually see good glucose stimulation, insulin secretion results.
Great.
It's not, I should be clear, I guess we get questions about, is this useful for transplants? And that's not really the goal of this platform. It would have to be reconfigured to do that. Really, this is meant to be a drug discovery or drug testing platform.
Yeah, it's like a window.
Exactly right.
Biological window, right?
Exactly right. So yeah, so this platform really has three main components. So we have a central cell chamber, and it's flanked by what we refer to as the arterial channel and the venual channel. And the reason we refer to them that way is that when we inject in cells, we put a hydrogel with endothelial cells and stromal cells in the middle here. But to actually get those to form a blood vessel network, we'll put a high pressure side. So again, arterial side of a capillary bed is going to be high pressure. And we're going to drive a blood-like substitute to at first diffuse through that hydrogel and then out the low pressure venule side. Now, of course, blood vessel networks form over time. And once those form, about 5 or 6 days later, you get a blood vessel network, and then your blood-like substitute is actually flowing through this platform. So a lot of microfluidics platforms use pumps and tubes, and they can be quite complex. And when we first developed this, we actually aimed to make it quite simple that it could use in any lab, anywhere. So we've got these medium reservoirs. So you can see here, this is the chamber here. This is the top channel and the bottom channel. And it's all gravity fed. So what's really cool about this is that we can just top up different levels of medium in each of those such that we can maintain this high pressure to low pressure flow for up to 2 days. At the end of the 2 days, you just top it up again and it keeps flowing. It's a pretty innovative solution that I can take no credit for developing.
No, it's really innovative. And I just wondered, are you guys looking at both, nod mouse islets, rat islets, human islets? What have you got?
Yeah, we talked about that early on, and we decided we just wanted to do human. So everything in our system is human. So it's human endothelial cells, human stromal cells. There is some FBS that we can't do without, but otherwise everything is human. So that carries over to the cadaveric islets as well.
Right.
We did that intentionally. If you want a humanized mouse model to study diabetes, there are many of them out there. And I would argue that the immune system in a mouse is not the same as a human in some very critical ways. So for us, if we were going to model type 1 diabetes relevant to human patients, we wanted it to be all human in here.
Great.
So the really cool part about this is that, we inject the cells. You can see here on the left that they start as single cells at day 0. And after about 4 or 5 days, we get this nice luminized blood vessel network.
That's
and it'll continue to prune and modify and regrow and whatnot for, again, up to 2 or 3 weeks. We can keep these out. We do put fluorescent dye through, so you can see that here, a rhodamine dextran. And what I want you to appreciate here is that all the dextran is going through the blood vessels. Now, we chose 70 kilodalton here because that's about the same size as albumin, a key component in blood. But we've tested this with other sizes as well that would be more indicative of small molecules or even larger, like antibodies, closer to 150 kilodaltons. My colleague got this great video here of fluorescent beads going through the system. This is kind of how immune cells or blood cells are going to flow through the system. So if you actually were to put a side-by-side of a capillary network in the body alongside this, you see kind of some cells that stop, some that move a little faster, some that move a little slower. This is very characteristic of an in vivo capillary bed.
And Hugh, are those for the far right image? Yeah. Are those beads that are particles that are going through about the same size as a T cell, a blood cell?
They're larger than a T cell, I believe. So I think these were, if I recall, 15 micron beads, 10 or 15 micron beads. So they're a little larger than what you'd expect for immune cell. We have some videos, I don't have them to share today, but of immune cells going through. And yeah, T cells are just so tiny, they just kind of whiz through there until they adhere and extravasate.
But if a bigger thing can go through, then that really proves the smaller definitely can make it too.
Exactly right. Exactly right. So we talked about getting enough islets into the device. And what I showed you there was that diamond chamber, which we use for a lot of different applications in our lab. But for the islets, it just really was important early on to put in many islets in there. So we put about 15 to 20 on average in there. And we get these really beautiful blood vessel networks. So just to orient you here, the endothelial cells are in red. Our stromal cells, which we don't always label, you didn't see them in the previous pictures, they're in the cyan color here. yeah, I think you can probably appreciate in some of these areas, they're actually wrapping around the blood vessels just like they do in the body. And that's partly why we don't have a lot of leak from these blood vessels. And then in the white, you can see the laminin basement membrane, again, very characteristic of the vascular bed, and also characteristic of that low leak type of environment. So we put islets in here. This is actually the same device right here after loading. So we put the islets in actually at the same time as we do the endothelial cells and stromal cells. So the vascular network actually kind of co-develops with the islets in there. You can see this is, I think, about a week in, and the blood vessels have formed all around the islets here, as outlined with these dashed lines. What was really critical to us was that these blood vessels are actually close enough to have interactions with the islets. So we got some really nice confocal images here. Again, on the left, our nuclei are blue, blood vessels in red. You can pick out the islet with this cluster of small nuclei here. This is reproducible. I mean, we just get a lot of endothelial cells that just go right next to the islet, wrap around them. And we think that's critical for actually getting the nutrients to the islet and, again, removing the insulin that they spit out.
Does anyone,
go ahead.
Hugh, does anyone know what the guiding molecule is that sort of guides those, the vasculature towards the beta cells or islets?
Yeah, it's a great question. We think islets have a lot of VEGF expression. And so we think that they're actually maintaining the VEGF expression throughout the process, actually, of isolation and everything, because you'll often see these luminized segments actually form before they form anywhere else. And then they kind of, you see others migrate down and join up. So we're pretty sure it's VEGF.
Yeah.
We haven't done a careful study of that, but it's kind of well known in the field. We've also done some more recent studies looking at kind of the environment that the islets are living in. So here the islets are in yellow. We see 3 of them here, various sizes. Again, the endothelial cells, the blood vessels in red. And what we're seeing is actually not all the islets, but a lot of them are actually regenerating these laminate basement membranes. So these are a key hallmark of islets in the native pancreas. So we think that's a key component to maintaining these islets, the health of these islets in the platform. So of course, the proof is in the pudding. If you can't get insulin secretion out of these islets, you have a nice, pretty model, but it's not necessarily very functional. So the way we test this is we'll put low and high concentrations of glucose in one of these reservoirs. We'll flow it through. It'll go through the blood vessel network, stimulate those islets, and we'll actually be able to measure insulin that comes out the other end. We do this at 10-minute intervals. That's kind of, again, I have to stress this is a very small platform. So we're talking about fractions of a microliter per minute. And ELISAs do, unfortunately, require a lot of volume. So we've gotten it down to about 10-minute intervals. And what's cool about this is that, just to orient you here, so we got the blue, that's our low glucose, our green, that's our stimulatory glucose concentration. And then we do test the machinery of the islets, seeing that they are in fact capable of secreting insulin with a potassium chloride solution. So in the black line here, you can see our glucose trace. So we do actually measure the glucose that flows through there. Again, it's on the right axis here. You can see it's kind of going from 5 to 16.7, exactly as we would expect. What's interesting to us is that you get this kind of gradual increase of insulin secretion. So I think the field is probably very used to seeing perfusion assays where you kind of bathe the islets in a lot of stimulatory medium and they spit out a whole bunch of insulin that you can measure. And so as a result, you see this steep peak and steep drop off. So it was a little head scratching to us why that was the case, that we see this kind of gradual increase in glucose, gradual increase in insulin, and again, gradual decrease. And so we actually, there's some, I guess not so recent data anymore, but looking at glucose traces and insulin traces post-meal in human patients. And, you know, I have to stress again, our platform is small, so the time scale is different here. But the overall shape of the curve is very similar to what we see in vivo. So this is probably something we're going to have to convince the field of, that you're not looking at something like a perfusion assay, you're actually looking at something much more gradual and fine-tuned that you would see in the body.
Yeah, and potentially titratable.
Exactly.
Yeah, I mean, yeah, it's right. The challenge will be to scale this system, but if you can scale it, it could be really a powerful system.
Exactly right. Yeah. So lastly, this is my last slide. I'll just talk a little bit about the type 1 diabetes modeling we're doing. So everything in our system, it's not donor matched. So we have a non-autologous system. That's kind of just the nature of where we are right now and where the field is right now. So your islets are always going to be mismatched from endothelial cells, which are going to be mismatched from your immune cells that you put in. But it was important to us that we kind of show a proof of principle that you could do type 1 diabetes modeling. So what we've done is we've taken peripheral blood mononuclear cells. So this is going to be a whole mix of immune cells. And we put them in with donor islets in these round bottom wells, so they have close association. On the left here, we've put them in with MHC blocking antibodies so that we're just kind of controlling for any MHC activation. On the right, you can see that these are the same immune cells with the same islets, and you see these characteristic grape-like clusters. So these are indicative of clonal T cell expansions. We let these go for about 5 or 6 days, and then just as a proof of principle, we've taken those same PBMCs and we put them on a new set of islets from the same donor. On the left side, you can really see the islets still look reasonably healthy. This is about after 2 days of being exposed to the immune cells. On the right, I hope you can appreciate on the white arrowheads, we're starting to see a lot of these pock marks and morphological damage that we're seeing to these islets. And we do think that's due to the activated immune cells going through there and attacking them.
Yeah.
So this is a static assay. Oh, sorry, go ahead. Do you have a question?
No, I was just going to say, yeah. So like pockets of apoptosis.
Yeah, we think so. It's kind of interesting that it's very focal. It wasn't necessarily what we would have expected. I kind of would have expected to see the whole islet just break apart. But the vast majority, I mean, you see that a little bit here on the right with this other islet, but the vast majority are still intact. You just see damage in focal areas.
And when you like, if you, sort of analyze this further, do you see that those pockets of damage are specific to beta cells?
Yeah, so we haven't done that. We've been looking at cell death in general just to see if that is actually correlating. It does seem to correlate in that area. We haven't looked at the beta cells. The beta cells, as you may know, in human islets are kind of spread throughout. They're not like a core as there are in mouse islets. So locationally, my guess is that we are getting a lot of beta cells. It's possible damaging some of the alpha and delta cells as well, which would be interesting in its own right.
Yeah, and it's a patchy, attack too, just like
exactly
in
right.
real life in human in vivo.
Exactly right. So yeah, this was kind of all a proof of principle. Can we actually kill islets this way? But of course, the most interesting thing that we want to do is put it actually back in our Islet VMO platform. So again, we've taken these same ones. The islets are from the same donor that we've activated them to. We put MHC blocked PBMCs through here. You see what you would expect to see in the body: very low adhesion, very low extravasation. Maybe we have one cell that's extravasating. Certainly no migration towards those islets that are in the dashed areas here. It's a completely different story when you put the activated PBMCs in. They readily adhere to the vessels, they start extravasating, and I can point out at least 3 of the 4 islets here have immune cells in them. It's just a blow up of that panel on the right. Yellow arrowheads here, you've seen a lot of these immune cells that have migrated out of the vessels. They're migrating towards the vicinity of the islet. And then in this, I counted at least probably about 10 immune cells that were in this islet. It's not all of them, so we're still investigating what the cause of that is, why we're seeing some versus others. Is it related to how close they are to the vessels? Is it related to something else? But we think this is compelling evidence that we can study type 1 diabetes in our platform. And so I'll just touch on some of the ongoing work we and our collaborators are working on. We're trying to use iPS-derived endothelial cells and beta cells to generate the same platform, but now everything will be genotype matched, so much closer to what you would see in the body.
Great idea.
Yeah. So that's all I have for my slides. I'd be happy to take any questions anybody has.
Yeah, for sure. I would love to open it up to questions. If we can just stop sharing and just go back to you.
Sure, yeah.
I see a couple people. I'm sure you know some of these folks. Rachel
I do, yes.
is here, and she's also just had her own paper come out. Congratulations again,
congratulations.
Rachel.
Thanks. Yeah, I was actually wanted to ask about your perfusion data, because we also were able to do some perfusion data in our work. And for us, we were able to measure, because we have a larger system that has more volume, which you don't have the courtesy of. We were able to measure insulin every 2 minutes right after that first high. So I was thinking, is it possible that since you can only get a measurement every 10 minutes, that's why it looks more gradual and you're not able to see that spike as much that you can maybe get in perfusion systems, because it's averaging out and so it looks more gradual?
Yeah, that's interesting. I mean, I think it's probably that delay is more just the time it takes to go and diffuse through the vessels and into the islets. We haven't seen any indication of a big pop there of an insulin secretion. That said, we are working with some collaborators at Florida State who have a pretty novel system that we'll be able to hook this one up and just flow media directly from our platforms and measure the insulin secretion in real time. We're looking forward to that, just for this plain and simple reason that it's less ELISAs, less glucose sample collection.
If I can jump in, who are those collaborators at Florida State?
Sure. So it's Mike Roper's lab at Florida State.
Yep.
So they do an insulin and isotropy system. They could tell you much more about the physics of why it works. It's basically a competitive assay. So you put in bound and unbound insulin and you can test the differences there. So we're looking forward to testing that. The machine actually just arrived in our lab a couple weeks ago, and now we're learning how to use it. So I'll let you know if we actually start seeing some of those peaks, but we, at this point in time, don't anticipate. We think this is a real result.
Well, I also think it's great that you actually measured glucose levels as well, because I think most people in the field just say we put Krebs buffer and it's this amount, but we don't actually measure it. So do you use like a YSI machine for actually measuring it, or how do you actually do that?
No, we use a much simpler system. We just use a glucometer that you can get at CVS. And it doesn't require a whole lot of volume, and it's pretty accurate. So.
Yeah, because for some reason, glucose meters have never worked for me with just cell culture media. And I thought it was because glucose meters are usually account for hematocrit, which media doesn't really have, but I'm glad you were able to get it to work. I didn't have it.
Yeah, it matches up with our calculated values as well. So yeah, if you ever find yourself doing that, I can recommend at least one that's worked well for us. That was actually from our collaborators down there. Like, oh yeah, we just use a glucose meter, which is much cheaper and easier than other alternatives out there.
Well, you know what Einstein says, science should be as simple as possible and not any simpler, something like that.
Yeah.
Right.
Yeah.
So yeah,
we try to simplify the things we need to. Waste our time on the more complex problems.
Because there's plenty of them.
Yeah, that's
Is there anyone else from the audience who'd like to ask Hugh a question? I guess I will ask just a sort of a quick question. This really interesting paper that just came out a couple of days ago from George Gall et al. It was in Cells, he's at Leipzig University in Germany, sort of titled, it was titled Epigenetic Drifts During Long-Term Intestinal Organoid Culture. I know you guys are all on to 3D organoid culture, and that's kind of an interesting idea, right? That like as you're placing the organoid into the long-term culture, maybe some DNA methylation or demethylation, sorry, gets involved and that ceases, or interplays with the metabolic adaptation. It's kind of a curious thing I hadn't really seen in the literature before. I don't know if you had.
No, I mean, it's interesting, but in some ways it's not surprising. I mean, we think about, think about growing cancer cell lines, right?
Yeah.
They change over time in just tissue culture. I think we would all like to believe that things don't change when they're in a 3D system, that we're now taking them off those harsh plastic petri dishes and everything. So it should be more native and better. But I actually take those data, I haven't seen this paper, but I actually take that as a positive. So we've have some data that, so we've done some single cell sequencing in not my platform, but in other platforms. And you can actually look at the signature of endothelial cells and just when they're on their own, so just blood vessels alone, versus if they're with colon tumors.
Mm-hmm.
And what's interesting is that the endothelial cells actually adopt a phenotype that is much more characteristic of these kind of torturous endothelial cells within the tumors themselves. So we do know that, it's a biological system. There's a lot of crosstalk. We do know that they're kind of all molding the different cell types into the types they want them to be. So I think that's a positive. I'd have to go look at this paper and see if they're framing it that way. Maybe it's not.
No, it's just sort of food for thought. And I would also just comment that wouldn't it be an interesting experiment to look at the vascularization in your 3D organoid system prior to immune attack, if you want to call it, and then after immune attack, like what are there changes in the actual endothelial cells, just as you're talking about there is in the as it intersects with a cancer cell. So like as things go south for the islets, what happens to the endothelial cells? That'd be a cool experiment.
Yeah, I mean, we're also quite keen right now to look at the effect of inflammatory cytokines. There's some debate about, there's a chicken and egg situation with diabetes in general. Is it the beta cells that are triggering the immune cells to attack them? Is it just rogue immune cells that have decided to attack the islets? Is it something in between? We think our system is going to be uniquely positioned to actually look at those interactions and kind of see who's the bad guy in the system, I guess. Mm-hmm. But that's one of the things we really want to look at. It's the work we're doing right now is to, if you put in inflammatory cytokines that are upregulated in type 1 diabetes patients, are we seeing, damage to the vessels? We expect a lot of it to be damage to the vessels, increased leak. Are we damaging the islets and their ability to respond to glucose? And perhaps more importantly, are we hyperactivating these immune cells that we're putting through? So it's a biological system. There's a lot of moving parts, but I think we're going to get some really interesting answers there.
Yeah, there's a lot of work right now interfacing the microbiome's impact, right, on a potential impact on islets as well, and whether or not they have any, negative role on the sort of development of type 1 diabetes. Would it be cool to have two of your systems, one sort of microbiome based, and then the other one with sort of feeding in to the to your islet scaffold? Be cool.
Yeah, it's I think that's what everybody talks about, the body on a chip. And I think when the Department of Defense first started funding these platforms about 12 years ago now, that's what they wanted. They wanted to just basically have Lego pieces. Okay, we have a brain, we have a heart, we have a lung, we have a liver, and now we can put these drugs through and watch it get processed in the liver, go attack heart cells.
Not going to be that simple, but
yeah, that's turned out to be quite difficult because while we have a universal medium called blood, turns out all our systems are very picky about what types of medium they're dealing with. So I think everybody's kind of still working on what is the universal medium, since it's still frowned upon, I think, to put the whole blood through these platforms.
Is there a repository for blood of newly diagnosed patients? Is one developing? I heard sort of a rumor.
NIDDK has been doing this at the national level, kind of to the idea that you could start doing IPS derived derivatives of this. So driving from immune cells in the blood and turning them into islets and endothelial cells. Certainly much easier to turn them into endothelial cells. Islets are still quite tricky for everybody in the field. But as a result of that, I mean, I think there are 4 different groups in our consortium that are working on patient-derived cells. And as a result, NIDDK is, I think, beefing up their repository of patient blood.
Yeah, that's the word on the street. That's great news.
Yeah.
Well, it was fantastic talking with you. Do you have, you're still planning to stay in Irvine, or what's next for you?
Yeah, so I'll be at some point, once we finish this paper, I'll be unfortunately moving on to the commercial side. So I've enjoyed my time in the lab, but there's a lot of work to be done on commercializing this.
Yeah,
not this specific platform,
news.
but
yeah, I think that's fantastic news.
Yeah.
Bring all that you've learned in the bench to industry, and and bring it to the clinic.
Exactly right. Yeah, that's what our goal is. So
it's great talking to you today, Hugh. Thank you so much for joining us.
And thank you for the invitation. I enjoyed talking.
Yeah.